welcome, Dr. Sartor. We're really happy to have you here. Dr. Sartor is a distinguished professor of medicine at the University of North Carolina, and he has published prolifically, and we're really thrilled to have him here today. Go ahead, Ildiko.
Thank you, Annabelle. Thank you, Dr. Sartor, for being here. We really are very excited to talk to you. And I know you have some introductory thoughts you would like to share with the group, and then we will, I'm sure, ask you some further questions. But please, I would love for you to introduce yourself and your research and what your thoughts are about Crohn's disease.
Yeah, well, thanks to everyone for organizing this. It's a tremendous opportunity, I think, to talk on my favorite topic. And I'll give you some insights as to why it's my favorite topic in just a second. So I've been at the university, I'm easy to introduce because I've been at the University of North Carolina since my GI fellowship. I do show a date here that'll tip things off, but for a while, let's say. I am a practicing gastroenterologist with a emphasis on IBD management, particularly Crohn's disease. I started the IBD clinic, multidisciplinary IBD clinic here at UNC that now has 7 adult gastroenterologists totally exclusively treating IBD patients and 3 pediatric gastroenterologists and then surgeons, pathologists, et cetera. So it's a pretty good sized group. So I have recently stepped back from my primary clinical not primary, but my clinical activities to spend more time in the lab. I run a pretty good size NIH-funded laboratory and have been interested in how bacteria drive these diseases, and importantly, what can you do to interrupt that cycle in patients that have disease? And I wanted to share a few introductory slides that will bring up concepts that I think will put everybody on the same wavelength, and then hopefully have a free-ranging discussion. If anyone has questions as I'm going along, please feel free to break in and just say, what do you mean by that? Because physicians tend to throw terms that are not lay friendly, and I'll try to do my best. So let me see if I can share the screen. Okay, so what I thought, I've got kind of a grandiose title here, concept of Crohn's disease, pathogenesis, and novel approaches to sustained remission without prolonged immunosuppression. I would love to put cure there, but we got to get into a sustained remission first. So first of all, why am I interested in Crohn's disease? Well, it's a vested interest. I developed Crohn's disease as a senior in college. I was at Washington Lee University in Virginia, and was diagnosed with something I had never heard of. They called it ileitis back then, but it's Crohn's disease more globally. And so my career really, I initially was going to be a GI surgeon based on some experience I was an orderly in an operating room in Shreveport, Louisiana, where I grew up during summer breaks in college. But the first insight I had was I was pretty sick as an intern. Spent 5 weeks in the hospital, and that's a long time to think. And so the first insight was actually from my hospital bed. And it occurred to me, I was really frustrated with the lack of therapies that work, at least for me. The epiphany was that we're highly unlikely to cure a disease unless we understand its causes. And I do think there are multiple causes of Crohn's because it's a very heterogeneous disease, meaning that some people have one presentation and others have other presentations, as simple as location, small intestine, ileum versus large intestine. The later insight was as a GI fellow, and I was actually at the beach in 1978. And that epiphany was, it's the bacteria, stupid. I had just read an article that talked about how bacterial cell walls, fragments of bacteria, could produce chronic inflammation in a rheumatoid arthritis model. And it struck me that I had always wondered why the ileum, why the colon. And it struck me that it's the bacteria, stupid. And that really propelled me into doing my first research. I've never done research. I went through medical school in 3 years and at Baylor College of Medicine in Houston, and then a busy internship and residency. And I was entering fellowship and wanted to do research, but I've never done any. So that gave me a focus. And so let me give you my current ideas of what causes Crohn's disease. And so in my view, and this again is my personal view, chronic intestinal inflammation is due to an overly aggressive immune response to a subset of bacteria in our intestines in a susceptible individual. And that's a key point. Susceptible individually. This susceptibility is probably genetically determined, and why those people that have genetic susceptibility develop disease is probably due to environmental stimuli. And so the overreaching kind of way of putting this together is that there's genetic susceptibility, there are environmental triggers, there are bacteria and viruses, fungi in our intestine that lead to an overactive immune response. So the way this fits together is that genetic susceptibility is a key feature, but alone, it's not ability to, it doesn't necessarily predetermine that you're going to get disease irrevocably. Like cystic fibrosis, everybody who has that gene gets disease. But in Crohn's, even identical twins, only 50% of the time, if one twin has disease, will the other develop disease, somewhere between 40 and 50%. And so the environmental triggers, I think, are important to initiate a process in a susceptible individual that then is driven by the gut bacteria to stimulate an overactive immune response, again, in a genetically susceptible host. So environmental triggers happen in everybody, but in most people, there's healing. But in Crohn's disease, there's progression to a hyperactive immune response that I think is driven by the bacteria. And so I'm going to break these down a little bit. You guys probably know a lot about genes, so I'll answer questions, but not emphasize that right now. So environmental triggers, I think, can be divided into two components: those that alter the gut bacteria, like antibiotics and diet, or alter microbial function. Diet certainly changes metabolism of the bacteria. And also those factors that alter the mucosal barrier, the lining of the intestine that keeps the bacteria out, can be broken by non-steroidal anti-inflammatory drugs, aspirin, Motrin, ibuprofen, infections, and smoking and stress certainly alter barrier function and immunoregulation. And so these factors kind of determine who with genetic susceptibility encounters environmental factors that initiate the process and may keep it going. So one of the most obvious factors, and one that I think is amenable to treatment, is diet. And there's a lot of epidemiologic evidence that in both Crohn's and ulcerative colitis, that high intakes of fat, omega-6 fatty acids, which are the harmful fatty acids, red meat, processed meats, have an increased risk. And that high fiber, fruit, and vegetable intake is associated with less of a risk. Now, these are just associations by epidemiology, and it remains to investigators to try to break down these factors. And so, in my view, one of the main effects of diet is to altering the composition and metabolism of gut bacteria. And we know from some work that I've done and work that many, many others have done, that growth and metabolic function of aggressive bacteria can be driven by refined sugars and iron. Protective bacteria can be promoted by eating, by interacting and metabolizing complex carbohydrates such as fiber and prebiotics. And so the idea is that you can potentially increase the number and the function of the protective bacteria by eating a healthy non-Western diet, but the Western diet that is evil, in my opinion, will promote the growth and metabolism of injurious bacteria. Okay, so what about the bacteria? So the reason this struck me at the beach in 1978 is important, because I'd been stumped by the conundrum of why does Crohn's disease happen at the end of the small intestine and in the colon, particularly the right colon? Well, we coexist with a huge number of bacteria, and they increase in complexity, both number and complexity, as you move from the relatively sterile stomach through the upper small intestine. But in the distal ileum, where Crohn's tends to happen in about 70% of patients, they strikingly increase, and they change in character from the so-called aerobic living with oxygen to a anaerobic flavor in the end of the small intestine. And then they jump up even more, another 1,000 to 10,000 fold in the colon. And the right colon, which Crohn's predominates, is an area of relative stagnation. It doesn't move as fast out of this side than on the left side. So everybody on this call and everybody in the world has a very unique composition of bacteria, viruses, and fungi, in aggregate called the microbiota. And this is determined in an individual by host genetic factors to some extent, but to a much greater extent, environmental factors. And we have talked about diet, infections, antibiotics, all of which can either positively or negatively influence the health of the bacteria. Now, in a healthy individual, a normal individual, the bacteria are characterized by high diversity. There's a huge number of different bacterial species were dominated by protective bacteria. And these bacteria live in the ileum and colonic so-called lumen, the inside of the gut, but they're excluded from the immune response by an effective protective barrier called the mucosa that consists of mucus as well as intact epithelial cells. And there are certain cells that sample this, the gut bacteria, the antigens, so-called, in the bacteria, that then stimulate a protective immune response. You may have read about regulatory T cells and B cells and macrophages, or Tregs for short, regulatory T cells. And IL-10 is something I will mention in just a second. But the net effect is we live in peaceful coexistence in the normal state with our bacteria. They're excluded, they're good bacteria, they help our immune system stay healthy, and the healthy immune response can then attack a invading pathogenic bacteria like a salmonella or a virus that causes dysentery. So in contrast, in Crohn's disease, the bacteria are less diverse. They are dominated by aggressive bacteria. And moreover, as I mentioned, there can be defects, either genetic or acquired. This is where the environmental triggers come in, that break that mucosal barrier, allow these aggressive bacteria to then stimulate aggressive immune responses. And it becomes a self-perpetuating factor, because the products of these immune cells further damage and ulcerate the mucosa. And so then you have unrestricted uptake of these aggressive bacteria and fungi that then have a field day stimulating the immune response. And so Crohn's disease, as I mentioned, is an activated inflammatory response and with mucosal ulceration. So this is the way I put things together. Now, the evidence for this is that there was a great study done under support of the Crohn's and Colitis Foundation in new onset pediatric Crohn's disease patients. They took patients at the time of diagnosis, got stool specimens, took biopsies, and they measured what bacteria are there. And the middle line is normal, okay? So some bacteria are increased, Enterobacteriaceae, Fusobacteria. These are E. coli, and Fusobacteria and other bacteria are expanded. And other bacteria, such as Lactospiraceae, Clostridia, Bifidobacterium, are decreased. And these turn out to be the protective bacteria that are decreased. So you have an abnormal balance of these bacteria. Now, the trouble with these type of studies is that you know what's there, but there's no proof that they cause disease. They could be a result of the disease. You know, maybe Crohn's patients, inflamed environment, they grow more E. coli, so you know what's there, but you can't tell, are they causing the problem? And so the way to go about that we have taken is to look at microbial function. And I run a national resource that's funded by the NIH and the Crohn's and Colitis Foundation called the National Notabiotic Rodent Resource Center. And what we do here is we have these isolators that are completely sterile, and you can cesarean derive mice sterilely into this, and they live their entire lives and then reproduce in a so-called germ-free or sterile environment. No bacteria, no viruses, no fungi. And you can compare the activities of these mice with those that are raised normally under conventional conditions, are specific pathogen free, no pathogens. And so you can compare what happens in a germ-free mouse to a conventional mouse. Or you can take a germ-free mouse and put in a single bacteria or a group of bacteria, or viruses, or fungi. Or what we've done recently is take a germ-free animal and transfer either a mouse or a human fecal group of bacteria, do a so-called fecal transplant into these mice. And I call those humanized mice, because we can put an IBD stool specimen, and then that mouse develops the same bacterial components and microbial components that the human has. So way back when, early back in the 90s, we showed that bacteria were actually very important in multiple genetically susceptible mouse, rat, guinea pig, non-human primate. I didn't do the guinea pig or primate, but others have. And showed that in the absence of bacteria, there's no immune activation and no colitis, even these are genetically susceptible mice. IL-10 is that protective immune factor. If you have mice deficient in this, they can develop disease in the presence of bacteria, but not in the absence of bacteria. And if you take this genetically susceptible mouse, germ-free, put in resident certain bacteria, so say you transfer feces from a normal mouse, within a week you develop immune activation and colitis that expands over 4 weeks. We then showed, very importantly, in the 90s, that all bacteria aren't equal, that a complex of unfractionated bacteria from a rat would cause aggressive colitis in this germ-free, genetically susceptible animal. Bacteroides vulgatus, a single bacterial strain, induced moderate colitis. E. coli did not in this model, and the combination of cecal bacteria up here and protective bacteria would not develop colitis or minimal colitis. So all bacteria aren't equal. We showed also that the same bacteria put into different hosts would have different activities. This E. coli didn't cause disease in the rat, but did in the genetically susceptible mouse. And so I said that E. coli were expanded in IBD. It turns out that the strain of E. coli is very important. Back in the late 90s, up into early 2000, the French group demonstrated that adherent invasive E. coli were functionally abnormal E. coli. These invaded the epithelium cells. They persisted within one of the immune protective cells, the macrophage, and the macrophage couldn't kill them. They just sat there and induced immune responses. So then we took these examples of a human ileal adherent invasive E. coli and a mouse-derived E. coli that we developed, and showed that in an IL-10 genetically susceptible, IL-10 deficient genetically susceptible host, that you got different degrees of inflammation based on the type of E. coli. This is a laboratory strain that really essentially developed no disease. Human strain, moderate inflammation. But importantly, healthy hosts don't develop disease with these. So there is a genetic susceptibility factor, but there's a strain difference in different bacteria. And so the overarching concept is that there's a balance of protective and aggressive bacteria in each of us. And in the healthy environment, protective bacteria went out, and I'm listing some of these protective bacteria. However, in Crohn's disease patients, the pro-inflammatory bacteria went out, and I've listed some of them. Okay, so and an important concept is that this balance is different for every individual, and every individual responds differently to the bacteria. So I hate to burst the bubble of people thinking, oh, I can find one COVID-like virus or one COVID-like, salmonella-like bacteria that causes Crohn's disease in everyone. And that's probably not going to be the case, unfortunately. And that's what makes this a complex area. Now, if you believe that this balance is important, then it follows that altering this balance in an individual should be able to treat ongoing inflammation and potentially prevent onset or recurrence of Crohn's disease in high-risk host. So this is an area that I'm really actively thinking about now and investigating. So how do you go about correcting this so-called dysbiosis, this abnormal relationship of good and bad bacteria in IBD? Well, you can do a fecal transplant, and I'm sure everyone has read a lot about success and failures of fecal transplants. It's a mixed bag. Alternatively, that I think would be a better approach is to selectively remove the aggressive bacteria and enhance either the numbers or the functions of the protective bacteria. Or you can create a less hostile environment by altering the diet, modifying the diet, removing toxic metabolites, or metabolites that promote outgrowth of aggressive bacteria. I worked with a guy at Cornell Vet School, recently published a paper saying that certain metabolites in a Crohn's patient would stimulate the growth of these adherent invasive E. coli. And so how do we go about doing that? Well, I'm going to give you two examples of what we've done. We working with Yoshi Mishima and Aki Oka in my group, we took germ-free, genetically susceptible IL-10 knockout mice, put in human fecal bacteria. These mice developed colitis, clearly evident by 2 weeks. And after 2 weeks, we intervened by giving a mixture of 17 Clostridia, which are some of the protective bacteria, are a less complex group of 11 of the same strains. And we showed that wild-type mice, the healthy mice, didn't develop disease with the human stool. The susceptible mice, the knockout mice, developed aggressive thickening and inflammation of the gut. But both of the Clostridia groups showed protection, even though we started 2 weeks, when disease was active. And here you can see a blinded histologic score. Under the microscope, we grade the degree of inflammation, and it was significantly less. And so we're preparing this for publication as we speak. So I worked with a group in a company, biotech in North Carolina called Gusto Global. And they have I thought, a fascinating concept of let's replace the abnormal, the missing microbial functions that are protective in IBD patients. So what's missing is production of short chain fatty acids that are important for health of the epithelium and stimulating regulatory T cells, abnormal bile acid conversion, production of immunoregulatory stimulating microbial products, killing other bad bacteria, blocking uptake of iron, and stimulating promotion between that. And so they came up with a group of 11 bacteria that redundantly brought in these functions. And here again, we used the IL-10 knockouts colonized with human stools, waited 2 weeks, because I think therapy is much more relevant than prevention, and treated with the consortium of bacteria or control phosphate buffered saline. And we showed protection. Let's concentrate over here. Let me just get that out of the way. Concentrate over here, you can see that the inflammation score in the treated group was less than those untreated. So this was recently published last year. So how can we improve? How can we go past fecal transplant? Well, I think we can use a customized approach that selectively replaces missing components that either replace the beneficial bacteria or get rid of the aggressive bacteria. Concentrate on protective bacteria that normally live in the gut. So you put them in, they have a good chance of staying there. Refining fecal transplant to, make it a little bit more individual based. But I do think that the idea of using a refined bacteria that you know what you're putting in makes a lot more sense. And I think that we could really do much better on dietary approaches that would stimulate growth of beneficial bacteria and their beneficial metabolic activities, and target metabolic function and dominant antigens rather than the name of the bacteria. And implicit in here is a customized approach. Every patient may require a little bit different approach based on how their profile of bacteria looks. So current therapy, this is a little bit out of date, but I don't want to bash current therapies. Current therapies have been great. The biologic therapies, the anti-TNF, the ustekinumab, or that targets IL-12 and IL-23, and the new agents that are blocking IL-12 selectively have been, oral agents that block the JAK-STAT pathway have really improved outcomes in IBD patients. The problem is, once you start them, you have to keep taking them. And they all work by inhibiting the immune response. Well, that's great if you have an overactive immune response, but what if you encounter a pathogen? The problem with the immune function is there in our bodies to protect against invading bacteria, viruses, and fungi, and to surveil for surveillance and elimination of cancer cells. So the consequence of long-term immunosuppression is increased risk of infection and cancer, which is a real problem when you're taking, particularly if you have a child with IBD and you're faced with lifelong immunosuppression. So what I'd like to do, and this is my last slide, what I'd like to think about is how could we come up with a better approach? We may require steroids or biologic therapy to put the fire out, to get the overactive immune response back to normal. But maybe then, rather than continuing that for life, we have a much more physiologic approach that we stimulate the activity of the protective immune response that's defective in IBD patients by dietary or bacteria that we know will stimulate these protective immune responses. Restore the mucosal barrier. I told you how important it is to keep those bacteria away from the immune cells. Well, bacterial metabolites and dietary fiber, for example, can help stimulate these protective metabolites. And then finally, we could selectively eliminate those bacteria that are providing the constant immunologic stimulus by targeting the bad guy bacteria without the good guy. You'd have to be very selective in this approach. And so I think it's better to block bacterial binding, enhance killing, or to give beneficial bacteria that actually selectively decrease those protective bacteria. Both of those cocktails I showed you would decrease E. coli in the gut, for example. And so the idea here is to have a short-term use of immunosuppression, but then maintain with dietary and microbial manipulations that could be a much more physiologic, less toxic approach and not lead to long-term immunosuppression, which is the current strategy. And I think in the long term, this is going to be a much better approach. And so this remains, a goal, but we haven't yet achieved it. But that's basically where my research is trying to go, is to help achieve these goals by better understanding what dietary factors, what bacteria factors can be manipulated fairly easily in patients with a patient-specific approach that could lead to a long-term remission. So I've talked too long. I'd love to get questions. I do have a list of things that I'm doing now, but it'd probably be appropriate to open things up for questions, and then I can tell you some current activities if you want to.
I found your presentation really interesting. What I found, I really liked that first slide where you say it's bacteria, stupid. That was a good one, particularly since I think all of us here have this feeling too that this is something that's the cause. And so I'd like to know, I know that you had an article that you were recently or were part of, a long article about phages and how you were focusing certain phages towards these particular bacteria that have been linked to inflammation. Can you talk a little bit about that?
Yeah, I love the concept because, as I told you, if we could target the bacteria, particularly that are causing disease in an individual, we'd be way ahead of the game. The trouble with antibiotics are that pretty much every antibiotic to date is non-selective. It kills good bacteria as well as the bad bacteria. And so the conceptual advance of phages is that phages have a very targeted host. Bacteriophages live in everybody, and they reproduce by attacking a specific bacteria, inserting their DNA into the genome of that bacteria. Then they take over the machinery of that bacteria. They're like an invading alien, and they take over the machinery of that bacteria, and the bacteria dies, but then it releases a ton, hundreds more phages. And then they attack other bacteria. So you can be highly selective by doing that. Now, the trouble is that bacteria have evolved with phages. So they actually have ways of getting rid of the phages. So the trick with phage therapy, the concept's been around a while, but the trick is to try to eliminate the ability of that bacteria to kick the phage out and eliminate it. And the approach in that Cell paper, there were two papers that just came out on phages that I was part of. The really long one was the Cell one. What they tried to do was to come up with a cocktail of phages, and they had a really nice group in working with a group in Israel, and they had a very nice way of just mining the phage world for bacteria that was specifically hit Klebsiella, which is one of the Enterobacteriaceae related to E. coli that cause disease. And we're successful in some models, but not others, in eliminating those bacteria and decreasing inflammation. So I love the concept. Like all good concepts, it needs to be tuned in order to bring it to fruition and use for studies in patients. But the concept is great. The other great advantage is it does nothing detrimental to the immune system. So it's solely selected toward a very targeted bacterial species.
Hello, I'm back. My laptop is cooled. I'm inside, and my apologies for that little delay on my part. But I thought your presentation was absolutely excellent. It resonated with me completely, and very independently. In my layperson way, I've come to a lot of the same conclusions that I think you have reached just observing a child with IBD and asking a lot of the same questions that you have asked yourself from decades ago. So I want to start by stepping back and asking a very practical question. And my practical question is, when we are doing biopsies in the course of a colonoscopy on patients, on Crohn's patients, and you talked in your presentation a little bit about the importance of eventually personalization and the different bacterial, viral, fungal species that could be implicated in exacerbating or causing Crohn's disease. How can we, or why can't we, in the course of those biopsies, identify for an individual patient which more pro-inflammatory or culpable components of the microbiome may be present for us to target for that individual?
Yeah, well, that's precisely the strategy that one would have to do in order to personalize. But I think it could be simpler. All right, look, Ildico, right? Yeah, I think it could be even simpler because you could get a stool specimen that is much less invasive than biopsy. Now, a subset of the bacteria that live in that you can see in the feces will be present at the at the tissue level with the biopsy. But the advantage of a stool specimen is it's easily available. You can do it sequentially. You don't have to undergo a colonoscopy, which is a big deal. And particularly if you have to get to the ileum, that's you really have to have a full prep, and you can't do that sequentially. So you can take a baseline and then do an intervention and see if you're actually accomplishing what you're doing. It's not only finding the bad guys, but it's seeing what good guys are there, and even the metabolites. And in a fecal specimen, the other advantage of a fecal specimen is you can measure metabolites, and you can key in on what, I mean, we think we know, maybe the top 5, top 10 bacteria, the top 5, top 10 metabolites. And so what we need to do is to bring that out of the laboratory environment and bring it down to routine diagnostic work. And you can do that. Currently, it's a big deal. You have to extract DNA. You have to sequence it by several different strategies, and then you have this huge mass of data, and it requires a well-trained bioinformaticist to analyze it. Well, we know with COVID detection and things like that, you can do rapid, if you know what you're looking for, you can do rapid bedside diagnostic, or things that a normal laboratory could easily do by creating a plate of top 10, 15 bacteria and have different colors, present, high, low, non-existent, just like a COVID test. And that's what we need to do, in my opinion. And then have ready-made cocktails of bacteria, to replace abnormal profile A, B, C, maybe D, E, F, but start simple, and and then come in. So that's how we're going to personalize. That would be the path forward to conception of commercializing this and making it truly patient specific.
So I couldn't agree more. And I will tell you a little anecdote because it leads to a very direct and, again, very practical question for patients. So at diagnosis almost 6 years ago, I asked a very, I think, basic, stupid question, which was,
it's the stupid questions that are the most difficult to answer. There's no such thing as a stupid question.
And I said, why don't we do a baseline stool fecal microbiome sample profile, treatment naive, kind of baseline. And even if we don't understand exactly what to do with that information today, isn't that something that's going to be, perhaps useful 5 years from now or a number of years from now? Wouldn't it be great to have that information? And other than the blank stares that I received when I asked that question, with the caveat that there was a GI fellow in the room who was nodding vigorously, I think that the answer was, to your point, and you said it, outside of the context of the lab, of the research lab, there really is no way currently commercially to do a stool profile, a microbiome sample for patients or anybody else. So I actually, became so interested in this topic that I went and found the American Gut Project that you may be familiar with, and actually had them sequence my daughter's microbiome twice, once using the 16S RNA and once using shotgun. So same stool sample, 2 different methods. And I share this anecdote because the results were very different. So that's kind of point number 1. And in my discussions with them, and they were a really amazing group and incredibly informative, they educated me about a couple of things that I want to ask your perspective on. One is that to get really accurate profiling, you have to do a few over time, probably somewhere between 3 and 5, I was told, to get a relatively accurate sample. That the stool sample is not going to be identical to kind of the mucosal samples, another thing I was told at the time. And again, most importantly, we don't currently know enough to action this information. So my takeaway from all of this was diet, because I said, well, diet is something I can control. Diet is something where we can figure out, potentially ways to modulate the microbiome beneficially. But we still live in this kind of very fuzzy world where microbiome research is still in its relative infancy. And I guess what I'm trying to figure out is, I so believe in the potential of it. How do we move from potential to actionable clinical insights and therapeutic potential? Well, maybe that's a tough question. I'm sorry.
No, but I'm just saying that's exactly where I think the field has got to go. I laughed because Rob Knight, who started the American Gut Project, or I never can remember its name. I know him from a long time ago, and he came up with that 3 to 5 times, but based on an article we co-published, in which I got his help. He's one of those brilliant bioinformaticists that I was talking about, and before he moved to San Diego, he was at University of Colorado, and he analyzed a group of samples that we collected from. We had the idea that it'd be really interesting and potentially important to take families of Crohn's patients, in which one child had Crohn's disease, have a sibling without Crohn's disease, and both parents, because you do inherit your bacteria to some extent from your parents. And no one should feel guilty on this call if they have kids with IBD. It's not anything you did. But anyway, and then we did it. Then we measured sequentially. A colleague of Rob's at University of Washington University, St. Louis, said we should do it daily, but that really was onerous. So we tried daily, and patients revolted. And the patient was fine. The family members revolted. So we went to 2 or 3 times a week over 2 to 4 weeks. We started 4 weeks and came down to 2 weeks to make it simpler, and came up. Rob did this linear regression analysis and decided that at least 3 times was necessary to get a question, because there was day-to-day variation to some degree. Now, when you initially said you got 2 samples, a big variable there is if you got one before treatment and one after treatment, then the treatment itself can make a difference in the microbiome.
So it was post treatment, but both. So it was 1 sample post treatment, because it took me a few months to find the American Gut Project, but it was 1 sample, 2 methods. So 2 methods on the same sample yielding very different results, which suggested to me, how do we make sense of this information? Is it reliable?
Yeah, I mean, that's something we wrestle with in the field. Usually you get globally similar results, but the trouble with 16S is you can't get down to the species level identification, and with the metagenomics, depending on how deeply you sequence, you can even get to strain level differences. I like metagenomics. It's more costly and more difficult to analyze, but I like metagenomics because you get more information, deeper level of identification, and you also can look at the genes that are present in the bacteria, which you can't with 16S. And then you can correlate that with functional characteristics if particularly if you have metabolomics in the same specimen, where you can look at what metabolites are present. So we tend to gravitate now. 16S it's simpler, it's quicker. You can sequence larger numbers of patients at any given time, but the metagenomics gives you more information. So I prefer metagenomics for most uses. We still do 16S for a quick and dirty look.
Thank you. Thank you for just letting me get through my anecdote, because I found this so fascinating as a result of some of that trial and error on my part to understand.
But I think your strategy is exactly what I'm describing. Is okay, if we know in an individual patient what the bacterial profile looks like, and we still are in the infancy of appropriate bioinformatics of viruses and fungi. I'm not thinking at all they're not important. It's just that we don't have the wherewithal now to measure them as much. Getting better, but not there yet. But let's say you have an abnormal profile. Well, what can you do about it? And that's what I was trying to outline in that last slide. You can manipulate diet. You can manipulate, you can try to attack the bad guys or build up the good guys. And the diet is the thing that most patients and families can more easily modify. It's just that we have, if you ask the same people that looked at you blankly when you ask about the stool specimen, ask them what diet should you, and I'm sure you have, you probably got the response, well, avoid what bothers you. Well, that's not a very good answer. You do that anyway, and you do it whether they told you not to. Anyway, we got to do better, but we're getting much better. One of the things that I'm trying to do now is feed our either humanized mice or our selectively colonized mice, that we know what bacteria are there. If you give one food versus another food, how does it impact bacterial function, numbers, community structure, and function, most easily measured by metabolites. And it's clear that going back to that epidemiologic insight, that high fiber diets will stimulate the growth and the metabolism of beneficial bacteria. And that can only be a good thing. And the bad guy bacteria don't particularly like fiber. So you alter that relationship. Conversely, we've shown that a high sugar, a high fructose diet, a high iron diet negatively impacts that microbial function. And so guess what? Epidemiologically, that's what is associated with increased risk of IBD. I'm about to start a project with a guy in New York, Long Island, looking at pea protein. He showed in a mouse model that that plant protein, particularly pea protein, pea-derived protein, was beneficial. And I'm trying to replicate that in our humanized mice that have a more immune-mediated inflammation. So I think that the only way to get to practical application is to do basic studies that say, okay, if you give a particular diet to a host, in my case, mice, but eventually humans, here's what happened. Here's how you positively or negatively impact the bacterial composition based on our current knowledge. And here's how you positively or negatively impact metabolites in there. And if something looks really fruitful in a mouse model, I think it's open game to do a human trial. The beauty of diets is that you don't have to be FDA approved for a diet. You can really do it much quicker and more cheaply.
Agreed. Although it's a very complex area, and as you well know, it suffers from, I would say, lack of widespread recognition as a therapy, aside from exclusive enteral nutrition. I think because the evidence is also harder to get right. It's really hard to do diet.
It's because you got to do it thoughtfully. I mean, you can't just say, oh, patient gets better, worse. As a patient can feel better for worse, if they have a bad day. And so, you've got to have objective criteria. And I think at the same time, you have to look at fecal microbiota. You need to look at biochemical characteristics, metabolic characteristics in order to do it. And I would say the field has suffered from a lack of rigor because you have all kinds of publications in the scientific literature and outside the scientific literature of, this extract, this fad diet, this this herb will cure your Crohn's disease, and people making statements like that really hurts the field by going, the credibility of this field is zip. So
I think there are some, thankfully, more thoughtful, GIs who are now interested in and studying the role of diet and IBD, and I'm always grateful for that. I know we still need more data. I'm a big believer in it personally, and to your point, we don't need FDA approval, so it's something.
Yeah, but don't get me wrong. There's some premier investigators, notably University of Pennsylvania. Jim Lewis, Gary Wu are doing a really good job. There's a group of very careful investigators in Israel that are doing great jobs in pediatric IBD. There's some really good groups around the world, but as you're saying, enteral nutrition is exclusive enteral nutrition is about the only one that has come to pass. Jim has studied at Pennsylvania, studied the Mediterranean diet versus the Elaine Gottschalk's
specific carbohydrate diet.
Specific carbohydrate diet, which I would have predicted would be miserably bad, but Jim said it was equivalent, to the Mediterranean diet. So anyway, there's some very thoughtful people in the field, but they're relatively few and far between.
I have a bias in that we do the specific carbohydrate diet, so I'm a believer in it, but that's my bias, and I readily acknowledge that. But I did want to go back to your last slide, which I thought was so fantastic. And I mean, I thought all your slides were fantastic, but you really summarized it in that last slide of what are some of the potential therapeutic approaches? Because I think that's where patients want to get to, right. And you understand this, and certainly parents out there, we are very impatient, right? We want to be able to, in addition, perhaps to immunosuppression, hopefully someday, not have immunosuppression, but use more targeted kind of microbiome modulating therapies. So you talked about a number of them. Annabelle asked about phages, which is one. You talked about fecal transplant and antibiotics and prebiotics and probiotics. I'm hoping you can help me sort of see how we move forward with these in the future, because right now, I would say, and this is true for biologics as well, I would say every therapy has relatively modest has shown relatively modest degree of benefit in trials. That includes FMT, that includes prebiotics have been pretty underwhelming in the studies that I've looked at, but please correct me. Antibiotics, again, arguably beneficial over placebo, but again, still, none of these seem to be the answer. So the question is, I guess, twofold. One is that, are we averaging out, first of all, are we not optimizing these therapies? That's question number one. And maybe the prebiotic that you talked about, which is a compilation of a number of different organisms that are kind of rationally selected, is maybe a better optimization. Are we not optimizing, or does it go back to heterogeneity that you talked about earlier? Are we just really dealing with multiple different diseases, and maybe in one individual, it is really about the immune response and an IL-23, but maybe in somebody else, it could be the right antibiotic cocktail, or it could be the right diet, or it could be some combination of these things. Like, how do we get to figuring that out for patients?
All right, there are a lot of questions embedded in that.
Yes, there are. Sorry about that.
I think, yes, all the above. First of all, Crohn's disease is heterogeneous. And I've tried to make the point throughout that everybody's different, and that everybody will respond to the same bacteria in different ways. So it's not only the bacteria, but it's how the immune system is wired and educated. And so I'll try to break down some of the things I can remember, but I think, first of all, prebiotics and probiotics have been dismal failures, in my opinion. I'm not at all saying if you go to the grocery store and buy a probiotic, first of all, a prebiotic is a food that fosters the growth and metabolic activity of beneficial bacteria. So that's different than a probiotic. A probiotic is a beneficial microbe, usually bacteria. Saccharomyces cerevisiae is a fungus. But the trouble with current probiotics is twofold. Number one, they haven't really been well studied because the companies are trying to sell them for overall health benefits rather than, if it's disease specific, they have to get an FDA indication and approval. So they're not claiming health benefits for diseases. People read that into it, but health, this is healthy. The trouble is many of them come out of cultured milk, and they don't have staying power. There's a great study in Bologna, Italy, that took 8 different probiotics, VSL3. You may have read about this. And the VSL3 were primarily lactobacilli, a few bifidobacteria, and streptomyces, anyway, one other. And they took it twice daily for 9 months. Stopped 2 weeks later, not there. They're gone. They have no staying power. So my belief is we ought to be taking bacteria that normally live in the gut, and not only bifidobacteria that are many of the standard and lactobacilli mainly live in the proximal jejunum, small intestine. You can see them in the feces. They don't normally inhabit the colon. So that's not where the action is in Crohn's disease. It's the end of the small intestine, ileum, and the colon. So you want to get things that normally live in that environment. So that's the first caveat. The second is find ones that have functions that are protective in Crohn's patients. These so-called lactase lactic acid bacteria, the lactobacilli and the bifidobacteria, are present, are enhanced by drinking milk, and they're present in babies that are drinking milk. But pick ones that are deficient in IBD and then replace them. So it's an entirely different concept than the things that live in yogurt. So I think that's a practical way of doing it. And the two consortia that I mentioned that showed protection in the animal model, even in a therapeutic protocol, treating aggressive disease once it occurs, have those characteristics. They normally live in the gut. They're decreased in IBD patients. And the metabolites they make are decreased in IBD patients. Hence, target those for replacement. And back to your personalized medicine, you could target those that are missing in that particular patient. So I think that would be a quantum leap forward. So what I'm saying is you're going to have a companion diagnostic test that would identify either at the time of diagnosis, which most clinicians are not thinking about, bacteria at that point, or somewhere along the way, what's missing? What do we need to replace? What is proliferating and totally out of bounds that we need to target?
How would it be delivered?
The probiotic.
Yeah.
The better
ones that you're talking about that are
better probiotic.
Right.
I think it could be delivered many ways. You probably want, rather than doing a fecal colonoscopy, fecal transplant type thing, you could easily lyophilize, meaning put into a dry powder, grow these bacteria. Most of them are anaerobes. They have to grow in an anaerobic chamber. Dry them down, put them into a capsule, take it, but you'd want to have a capsule that doesn't open in the stomach. So you'd probably want it just like some of the mesalamine agents, the 5-ASA agents that are targeted to open in the ileum and the colon, to have them open down there with a oidrugate coating, for example. My belief is you probably, even though I'm saying they're designed to live in the gut, ileum and colon, you probably want to be taking them on a regular basis, would be my guess. That's what we did in the mouse models. So I think they could be easily packaged for delivery.
So that makes sense to me, to have a diagnostic, to have perhaps a combination of different, very specific and individually selected strains of bacteria that you would take on a regular basis, and perhaps fungi. But I wanted to ask you another question and kind of move to a slightly different topic, is that do you believe that the microbiome also impacts your response to current therapies?
I think it very well could. But there are two elements there. One is, could it impact current therapies? And I think the answer is yes, and I'll give you an example. But maybe even perhaps equally importantly, could it predict response to a treatment? And I think there's potential there as well. This is still theoretically based, but there's good evidence. One of the big advancements in cancer is so-called immunotherapy. Rather than killing the immune response, you actually enhance the protective aspects of the immune response that are decreased in cancer. And there's great work by a lady named Jennifer Varga, Varga, at MD Anderson, showing that the response to immunotherapy is contingent upon the bacterial profile. And she could even predict to some extent high responders and low responders based on their microbial characteristics. And that's an immune-based therapy. I don't see why you couldn't do the same thing. And people are trying to do that. I talked about that risk. I didn't give it a name, but it's called the risk study that was done in pediatric Crohn's patients at the time of diagnosis. They did it was really well planned. This all happened 10 years ago, but now they're paying the fruit of that, getting the rewards of some hard work early on. And I helped them design that study. Not on the paper, but I was with the Crohn's and Colitis Foundation helping guide that project. And so basically what they did is they took that sample, both mucosal biopsies and fecal samples at the time of diagnosis, and they found that they could look at some of that bacterial profile and some of the immune characteristics, and together those two diagnostic so-called biomarkers would predict those patients that had complicated Crohn's disease versus easy to manage Crohn's disease, and to some extent predict outcomes of TNF therapy, which is the only biologic they had in high enough quantities, high enough numbers to really analyze. So I do think the potential is there. Do we have proof yet? No. Do we need to get proof? Absolutely yes. Because, think how much, now that we have 10 different agents, basically the way you get to the right therapy is you try the first one and then see if it works. And if it doesn't work, try the next one and go on down the list. Well, meanwhile, the patients develop complications, time has passed, agony has happened. Why not get to the right answer first? That's the whole goal of personalized management, is get the right drug at the right time. And I think, I don't know that bacteria alone are going to be the predictor, but I do think that would be one of the factors, along with immune predictors and perhaps genetic predictors, that would be put into the equation. I told you on that first slide, that kind of the third slide, the diagram, the Venn diagram of the 4 components. You probably need to get predictive factors from each of those 4 components in order to make sense of the process. I was just saying the easiest to modify are the environmental factors and the microbial factors.
And I think from a parent perspective, and perhaps patients too, I would imagine, I think what's frustrating is that we see this. I think it's very rational, and I think we all understand kind of where we need to go, but it doesn't seem to be where we're headed, because it's a lot easier just to frankly take IBD patients, Crohn's and UC, and use the same therapies, and to your point, use trial and error. It's, I'm in the pharmaceutical industry. I have been for 20 years. Personalized medicine is something we've been talking about for decades. And outside of cancer, we just really haven't seen it take hold. And so I'm really strongly advocating for that in IBD. But I think there has to be a sea change in kind of the mentality to really look at individual patients, investigate individual patients, the microbiome, their genetic profile, their immunological profile, to put the pieces of the puzzle together for each patient. I just obviously don't see that happening at the bedside yet. And I understand practically why. I think it's very challenging.
It's challenging, but you mentioned cancer has done it. And why would you treat a breast cancer that is estrogen receptor negative with an estrogen receptor inhibitor? I mean, it doesn't make any sense. It's not going to work. And guess what? It doesn't. So we just, as a field, need to have a concerted effort to put our minds together, get the different disciplines, the microbiologists, the Rob Knights of the world, talking to the immunologic silo people, and the genetics are off in their own completely different sphere. And the clinicians are doing studies in a vacuum, and we don't have the right coordination, in my opinion. It's not that we can't
agree with that more.
We can't crack it. It's not that we can't crack it. We just haven't yet because we haven't put the effort to do it. You could also say there's a numbers game, that there are probably 20 times more people working on cancer than IBD. But I just want to going back to the previous topic of, I forgot to mention that a study that I am currently doing is actually testing the hypothesis that I brought up in that last slide. And that is, if you use either a steroid or a biologic treatment short term, will using bacterial treatment, and here we're using the Gut 108 because that company and I got a grant from the Crohn's and Colitis Foundation. So would adjunctive therapy, concomitant therapy, be better than single therapy? So yes, we did attenuate disease with the Gut 108, but we didn't completely knock it out. With steroid therapy, which is widely used and 70-80% of people initially respond, you don't get mucosal healing, you don't get long-term responses. You stop it, and at least 50% of the time it comes back, usually higher. And so the idea is, if you could blend the two approaches, would you get, number one, a more rapid response? Number two, a deeper response? Would you get mucosal healing? And so we're actually testing that in our humanized mouse model. And we're doing, at the same time, we're looking at a biologic therapy that works to some extent in mice, but not completely. So we're using the anti-IL-12/23 approach because it works better in mice than anti-TNF. So to be determined. We'll know in 6 months whether that worked or not. And then the second phase of that project is, okay, if that works, can you then stop the immunosuppression and maintain the remission with the microbial modification? So that's phase 2 of that project, funded by the Crohn's and Colitis Foundation. So
that's excellent work, and
I'll give you an update in 6 months.
Thank you. I'm grateful to you all for doing that work, and for the Crohn's and Colitis Foundation to fund that. I think that's excellent. I think where we want to get to is when we talk about combination therapies, which seems to be a hot topic these days in the GI community, and it's often talked about in the context of dual biologics, combinations of biologics addressing multiple pathways. I, at least as a parent, would be much happier to talk about combination therapies where we're talking about, perhaps a temporary use of an immunosuppressant in combination with some sort of microbial modulation that then kind of avoids that long-term immunosuppression and the risks associated.
I completely agree with you, because as interesting and it appears some early results, combination therapy sounds may prove to be highly beneficial, it's going to double your chance of complications as well. Because if you're blocking TNF and anti-IL-12, P40 are blocking JAK pathway and IL-12 or TNF, then you're further damaging the ability of your immune response to fight a pathogen, and for surveillance of cancer and elimination of cancer cells. So it's basically going back to the cancer immunotherapy. That's the problem with the immune response in cancer, that cancer is smart enough that that it actually decreases the immune response and therefore can grow. And so immunotherapy is about enhancing that immune response, kill the bacteria, kill the cancer cells.
And that's always been a question of mine as well in IBD. There was a company, I don't know if you were familiar, called Q Biologics, QU Biologics in Canada and Vancouver. Right. And they were looking at doing essentially that, enhancing the immune response.
Yeah, and they showed some early. I haven't talked to them lately, but yeah, I've talked to them over the years, and it sounds paradoxical that if I say at my beginning that it's an overactive immune response to bacteria, I also I changed the wording of that. I said in a setting of immune dysfunction. So what they're trying to do is to activate the innate immune response, which is good evidence that some of the genetic defects are targeting bacterial killing, for example, and autophagy, for example. So the bottom line is, it sounds paradoxical, but if they have results, you got to pay attention. And I told them, you might stir things up, and I haven't seen the data. They tell me that it's very very positive, but I haven't seen the data.
Agreed.
And again, Chuck Elson at University of Alabama is doing a similar thing. He's trying to specifically enhance regulatory T cells by altering their metabolism, and he's got some very interesting data that was published in Gastroenterology, and I wrote an editorial about.
And actually, if it's the one I'm thinking of, it was last year. Is that right?
Yep, exactly.
That was an excellent editorial, and I'm glad you brought it up, because I wanted to mention that hopefully people can, if they're interested to learn more, can find that, can look it up. I believe it's open access, and so that is definitely worth a read. I thought that was excellent. It's really putting all of these pieces together about kind of how we move forward to try to be more thoughtful in our approach to Crohn's, rather than just sort of blunt immunosuppression. But it goes back, I think, to what you said on your first slide, which is we have to understand the disease if we want to treat it. And I think that was, you said something to that effect. I'm paraphrasing you, but that was my takeaway. And I think that's so right, is that sometimes we have a drug and we say, well, it works. The RCT, the trial that showed that it works. And it seems to me, just based on the complexity, that we really do need to unravel and unpack all of these issues to understand it better, if we really want to treat it better and have better efficacy and cures for patients.
Well, I mean, you paraphrased me quite well, I think, maybe a little more eloquently. But no, I mean, there are many ways to get to Crohn's disease, and ergo, there got to be many ways to treat it that vary depending on how you get there. If it's predominantly a genetic defect, you got to correct whatever that genetic defect is. If it's predominantly microbial overgrowth of bad guy bacteria, then you in theory, you need to correct that. Where knocking the immune response down in that situation may be a bad thing because those aggressive bacteria may grow even better. So I think we really have, and that's why I think a physician scientist does have a leg up over a PhD without patient interactions, in that you can be guided by the patient's perspective. I have the advantage of being a patient before I was a physician, and a physician before I was a scientist. So I've truly evolved in my understanding, but it still is grounded by, would I take that medicine? I'm not going to advise a patient to take a medicine if I myself would have questions. So we need to do a much better job of what's doing. I don't know if it'd be productive, but I could throw up a couple of slides of some projects that we're doing now, and I probably have overwhelmed everybody's capacity. And
not at all. And that everything you're saying is music to our ears, and I think incredibly important. And what I really appreciate about you, many things, but some of it is that you really were thinking about kind of the pathogenesis of disease early on, including from your hospital room, as you said, and really thinking about what the causes are, why there's heterogeneity, how we can treat better and safer. And I think all of those things really do resonate with parents and patients. I think it's incredibly important. And so I don't think you've overwhelmed us. I think you've given us really outstanding information. And I'm happy to look at those slides. And I also just want to ask Annabelle and Angela if they have any questions. But in the meantime, also please feel free, if you like to throw up those slides and tell us about those projects, we'd be happy to hear it.
Well, love to have other questions.
Well, I'm really happy with this conversation because it gives me a lot of hope, and I love listening to you because exactly what Ildiko said. It's interesting that we are. I feel like we're on the same page, absolutely. And trying to understand. I've never understood how we could not find a cure if we don't understand why it's happening in the first place, what the triggers are. So that definitely resonates. And yes, I think we would love to see your slides and see what you're working on, because.
Okay, well, I'm doing a lot of stuff. One of my downside weaknesses is I have difficulty saying no, and everything sounds interesting. So I have prioritized to some extent, but these are things that I've worked on in the past 5 years, and some that I've highlighted that I'm emphasizing moving forward. So first of all, why is the Western diet so bad? And I think, we talked about manipulating the diet, and each of these things I mentioned here have studied in animal models. I think it's important to have an animal model that's as close to human disease as possible. So we are spending a lot of effort putting IBD human donor feces back into mice, and we're getting very different responses from different donors. Again, going back to that heterogeneity of disease. We're looking at mechanisms of why is a protective bacteria protective? How does a beneficial bacteria stimulate a protective immune response? In theory, if you could better understand that, you wouldn't necessarily need the bacteria, but you could use the metabolite, or you could use an analog of that metabolite to stimulate protective immune responses. So, Ildiko, going back to your pharmaceutical background. I mean, if you know the target, you can, get your synthetic chemists to come up with some really cool approaches. And then an area that I'm actively looking at is identifying, isolating more protective and aggressive bacteria in healthy subjects. And we're putting together I spent the weekend putting together a manuscript that is basically showing that in a healthy individual, they're a composite of beneficial and detrimental bacteria, and we've identified a way to separate those and showed that the aggressive bacteria in a genetically susceptible host will induce disease, not in the healthy host. But on the other hand, the protective bacteria will not induce disease and actually reverse disease stimulated by the bad guy bacteria. So that's an NIH-funded project that I'm doing, and then, like we've just talked about, better ways to restore a healthy microbiota and functions to IBD patients by going through experimental colitis models to do a proof of principle and screen which ones might look particularly interesting. And that's rationally designed bacterial consortia using bacteria that normally live in the gut and are decreased in IBD patients, bacteriophages, blocking pathogenic bacterial attachments such as the adherent invasive E. coli, and how to block either the production or effects of toxic metabolites like hydrogen sulfide, for example. And then we just published a paper with a guy at Cornell Vet School on the inflammatory environment that stimulate overgrowth of disease-causing bacteria that I mentioned. Identify mechanisms of protection versus disease-causing bacterial metabolites. I'm really interested in the idea of bacterial cross-feeding. How does one bacteria influence another bacteria? In this case, by breaking down mucus to stimulate growth, provide products that the other bacteria can use to grow, because these are bacterial communities. Each bacteria doesn't live in isolation. It has neighbors, and they both fight with each other and help each other. And trying to understand that is an area that I've got 2 postdocs working on. And then predictors, biomarker, going back to the thesis of how can we predict outcomes. I'm particularly interested in postoperative recurrence of Crohn's, because if one could, right now we try to avoid surgery at all costs because it comes back. It may take care of a complication like a stricture or a fistula, but it almost always comes back. So how can we predict at the time of a resection or even before a resection whether that patient is likely to have a bad outcome? And then I've got a couple of projects going trying to predict the risk of postoperative recurrence in Crohn's tissues. So that's kind of the human translational component. So those are some of the things that we're trying to do, all consistent with the theme of how can we improve therapies and move it from mouse to man.
Well, that's also an incredibly important question, right? Is that, are you confident in the ability of these mouse models to predict effects in humans? That's all. I mean, that's always a question in any science, not just.
It is, and it's a key question because, if the woods are full of predictions from mouse models of therapies that don't work in humans. The problem is, my take on this is that you got to be really thoughtful. First of all, mice don't have Crohn's disease or ulcerative colitis. They have experimental ileitis or colitis. and it's not human disease. As I say, I'm trying to make them as human as possible with putting in the human bacteria, stack the deck. Secondly, I think it's really important to make sure that a potential therapy works in several different types of models, not just a single model. So one of the simplest colitis models is you have something called dextran sodium sulfate, DSS. The trouble is it's a mucosal injury model. It burns the epithelial cell. And you don't even need immune cells to get that colitis, as shown at University of Alabama. And so it's, to try to, and then third of all, you need to be able to reverse established inflammation rather than just prevent onset. And so the vast number of people that are screening, including drug companies, that are screening drugs are doing it in a DSS model in a prevention pathway, 5 days of treatment, 5 to 7 days of treatment, and then try to go to humans. Well, it's not a good prediction. So I think if you show it in an immune-based chronic model and you reverse established disease rather than just prevent, that you have a much better predictive value.
Thank you. That's very, very helpful. Annabelle, Angela, do you have any questions?
I don't, but I'm thrilled with everything you said here. Thank you so much for joining us and being part of this very good discussion.
Well, I just want to, before I hang up, just, thank y'all for the opportunity. And secondly, say that, I'm the cheerleader. I don't do these, I don't handle the mice or fish out the guts or the bacteria. I've got some incredibly dedicated, smart, young physician scientists, young PhDs working in my laboratory, as well as some folks that have been with me for a long, long time that actually get the job done. I just come in with crazy ideas and they make them work.
I wanted to thank you as well, Doctor.
Sometimes make it work.
No, I wanted to thank you as well for just the multifaceted way you've approached this. It really is just, it just blows me away with all the work and the approach you've taken in this. I have one little question from one of your themes of investigation about the recurrence of Crohn's postoperatively. Do you have any theory as to why so often it seems to be at that point of anastomosis and why no other place?
Yeah, well, I'm weird in that that's some of the things I think about at night. And my belief, and that's really an important question because you're right. It is almost always, the most common type of Crohn's surgery is take out the ileum and part of the colon, hook them back together. And that anastomosis, that hookup between the small bowel and colon is almost, I won't say universally, but 90% of the time, it's going to recur just on the ileal side of that. And I believe that's because you're removing the ileocecal valve and the colonic bacteria that I said are 1,000 to 10,000 times more frequent in the colon are now refluxing back into that distal ileum, so-called neo-terminal ileum. And so that area is exposed to far more bacteria than it normally might need to deal with. And that's what might accelerate. The other thing is that the anastomosis itself injures the mucosal barrier. And so you're opening the door right there for evil things to come in from the lumen and stimulate the immune response. One can go into great detail about immunologic and characteristics of the epithelial cell and ileum versus the colon that I don't want to bore y'all with. But there's one cell called the Panna cell that creates antimicrobial peptides that kill that, and those are present in the ileum and defective in many patients with Crohn's. So that's yet another pathogenic mechanism. But I do think it's probably the juxtaposition of haloids of bacteria where they're not typically supposed to be.
Thank you.
Thank you.
You can tell I'm a bacteriocentric person.
It makes so much sense. I mean, now that you talk about it, it's like, well, yeah.
Well, yeah. I'd have to tell you that 78 aha moment with its bacteria stupid led to a long swim upstream because it was at that point an autoimmune process. And there were a lot of people going, you are absolutely totally friggin' nuts.
And oftentimes those are the best people, those are the people who are often proven to be right. And of course, now everybody says, of course, the microbiome is implicated. Of course, this all makes sense. So that's why, as I said earlier, I'm so grateful to you for really being ahead of your time, I think, on microbiome, which is very hot right now, but to your point, was not very hot in 1978 or even in the 80s or 90s or early 2000s. So you were really ahead of your time in thinking about Crohn's in this way. I think it will make sense to a lot of people, and you've really given us a master class. So I just want to thank you so much for the excellent slides, for the thoughtful responses, and just for taking the time to speak with us today. I think this is going to be very informative.
Well, great questions, and like I say, I'm always happy to discuss things with a knowledgeable, enthusiastic audience. So it's been fantastic, and I certainly can email you these slides if you want to put them on a website.
Fantastic. Thank you so much.
Thank you so much. Really appreciate this.